This is the second part in a 4-part series hosted by the Climate and Agriculture Initiative. And over 4 weeks, we are discussing the ways that climate change is impacting BC farmers and ranchers. And our presentations are sharing different approaches, tools, and resources that provide examples of adaptation solutions in action. We are recording each session and we will be sharing them publicly. So if you can't make it to all 4 sessions and are still interested in the topics, you will have an opportunity to view those at any time after the sessions are finished. So my name is Harmony Bjarnason. I am the Regional Program Manager for the Climate and Agriculture Initiative, and I will be your host for today's session on monitoring and supporting pollinator populations in a changing climate. And before we begin our session to talk about pollinators' relationship with the land, I would like to acknowledge a relationship that goes as long and as deep. I am hosting this session from the ancestral and unceded territory of the Syilx Okanagan people, and specifically from the traditional lands of the West Bank First Nation. So thank you all for taking the time to join us. I invite you to introduce yourself in the chat, and I see that many of you are already doing that, so that's wonderful. Let us know where you're calling in from, why you've decided to join us today, and direct your messages to all panelists and attendees just so that everybody can see those. So just a bit of a session overview. For today, we have 2 new projects to introduce you to. Our session will be an hour long, and we will have 2 breaks for Q&A. So the presenters will do their presentation, and then we'll get into the Q&A right afterwards. To get us started, I will provide you with a quick background on the Climate and Agriculture Initiative and give a little context on how climate change is affecting pollinator populations and how we arrived to these 2 projects that are focused on this topic. And then we'll get into our speakers. I'm gonna move my slides over. Oh, hang on. Sorry, I don't know why that's not moving. There we go. So many of you who are joining us today are likely quite familiar with CAI. The Climate and Agriculture Initiative was established 13 years ago to provide a proactive and pan-agricultural approach to climate change in the province of BC. And our organization brings together other agricultural organizations, producers, government, researchers to collaboratively address climate change challenges through our work across the province. And we develop and implement projects and develop resources and tools to support agricultural adaptation to climate change. The bulk of our work falls into 2 programs, one that addresses regional issues and another one that focuses on farm level issues. And the projects you're hearing about today are part of our activities through those 2 programs. So the projects we deliver fall within the climate change adaptation programming of the Ministry of Agriculture, Food and Fisheries. And this funding for all the projects is provided by the provincial and federal governments through the Canadian Agricultural Partnership. We do receive additional project funding and in-kind support from many additional partners, including agriculture organizations, local governments, NGOs, individual producers, lots of collaborators, and you hear about some of those today. So just a quick overview on how climate change is affecting pollinator populations. And when we talk about pollinators, we're talking about bees and other insects such as moths and flies and wasps, as well as some birds and mammals. And pollinators, as you are all likely aware, play a very important role in seed and fruit production, and also have impacts on crop yield and quality on all agriculture produced across the province. And quite a number of changes in climate are likely affecting pollinator populations. So rising average annual temperatures across all seasons, including extremely hot temperatures in the summer, increasing growing degree days, so that's like heat accumulation in the environment over time, and growing season length. And shifting precipitation patterns, so increasing spring precipitation, increasing extreme precipitation events, as well as less precipitation in the summer, bringing on drier summer conditions. So these are just some of the changes affecting pollinator populations. And when we think about the effects of climate change on pollinator populations, we don't have a really great understanding of how all of these impacts are being felt and what the overall risk is. We know that warmer temperatures and extremes can increase pollinator mortality rates or reduce reproduction rates. Climate change may also affect pollinator plant interactions. So for example, plants may bloom at times when pollinators aren't active, or vice versa. There may not be enough floral resources available for pollinators when they're out foraging. And the combination of changes in precipitation and temperatures may narrow or change windows for pollinator activity. So without enough pollination, crop yields go down and edible plants have a harder time reproducing. BC is very rich in native pollinators, which play an important role in ecosystem health and crop pollination. But at the same time, all types of pollinators are under threat. So when we're talking about our pollinator projects today, we are thinking about the threats to both wild pollinators and commercially produced honeybees. So how did we get here? I'll just give you a little bit of background on that before I introduce our speakers. Pollinator health was raised by a concern by producers at a number of climate and agriculture initiatives, regional, through our climate adaptation planning processes. And it's really only been in the last year that we've been able to work with local partners in 2 of our regions to initiate projects in this area. So our work here is really just beginning, but certainly, understanding the threat that climate change poses to pollinators and what we can do to better manage that risk has come up across the province. And as noted above, there is much to be learned about the interaction between climate change, pollinators, and agriculture. But in many parts of BC, we really lack that baseline understanding about pollinator populations. And Aija White will speak to this. Aija and Bonnie will both speak a bit to this. Without that baseline, it's really hard to understand how climate change is impacting pollinators if we don't know what's there to start with. So we're really ultimately aiming to provide more and better information to producers, both about their regionally specific pollinator populations and about how they can actively support pollinator health and support a resilient pollinator population. You can visit the Pests and Pollinators page. It is on our website. That's the screenshot. If you go to our website, that's where you'll end up. And you can see a selection of curated resources and tools on this topic. So today's presentation, we do have 2 examples of projects that are gathering baseline data about pollinator health and moving towards providing more accurate and specific information to producers. And these projects demonstrate 2 quite different approaches to gathering data. And we hope that these can inform similar pollinator related work in other agricultural areas in the province. And these projects can also provide some food for thought in terms of methods for collecting data that is relevant to agriculture. Aija White is joining us from the University of Northern British Columbia to discuss the roles of pollinators in agriculture and the work that she's doing to quantify pollinator populations in the central interior. And Bonnie Zand is joining us again this week. She'll be sharing how she's engaging citizen science and using an iPhone app to better understand pollinator populations on Vancouver Island. So as I said, after each presenter, we will have a chance for a Q&A. You can use the Q&A box that you would like to see answered, and you can also upvote using the thumbs up icon beside the question. So that'll help us when we get to the Q&A to moderate those. So when you vote for it, it will rise to the top of the queue, and we can see that that's the question you want answered first. So with that, I will get into our first speaker. It is my pleasure to introduce Aija White. Aija is an ecologist currently finishing a PhD in natural resources and environmental studies at the University of Northern British Columbia. And she's investigating how resources affect life history trade-offs in mountain bluebirds. But today she will be talking about her work on pollinators. So over to you, Aija. I'm just going to stop sharing my screen and you will be able to put your presentation up there.
Does that look okay for everybody? Yeah, great. Okay. So yeah, thank you, Harmony. So I'm going to talk a little bit about this project that we've had going on since January, but we started monitoring in May. But what I want to do first is provide some background for why we might bother sampling pollinators and why everybody should be probably interested in this if they're working on the land. And it's because, you know, when I talk to people, oftentimes they think, why should I care about pollinators? I don't grow fruit or vegetables that require pollinators in order to produce. And the fact is that even for plants that are self-compatible and for plants that don't necessarily require insect pollinators, often your returns are way better when you have a nice diverse pollinator community around. And so I'm going to show you some work here from a couple of different studies, but there are lots more. These ones just had nice graphs that shows that when pollinators are present, yields generally are better, either in terms of, on the left, you can see a study from Stein and colleagues that shows that in cotton crops, when you allow pollinators to be present, the quality of the crop, so the fiber weight that you're getting out of the crop is better than when you just allow these plants to self-pollinate by a wind. On the right is an example from canola from Adaptation and colleagues. This is a greenhouse experiment, so highly controlled. And they showed that when you have pollinators present, generally you get higher yields. So it kind of makes the point that even if you don't think you need pollinators, pollinators are probably going to improve the quality and the quantity of crops. And furthermore, when we talk about pollinators, often people think I'm going out and counting bumblebees and honeybees, and that's it. And the fact is that we need to appreciate that honeybees and bumblebees, you know, the Apidae is the formal name for the family, are not the only pollinator, and they're not even the most effective ones sometimes, depending on the crops. From the left is a figure from a really nice review by Doyle and colleagues. And they looked at one alternative pollinator, so the family Syrphidae, which are dipterans, they're flies, and they're often quite mimic-like in terms of their appearance. They look a lot like bees. And they went out and they looked at a whole bunch of different studies and the number of plants that can be pollinated by these different groups of bees. So the Apidae, the sweat bees, which are the Halictidae, and the Syrphids, which are hoverflies, a kind of fly. And they found that hoverflies cover almost as many species of plants in terms of being effective pollinators as, you know, what we would traditionally think of as the honeybees and the bumblebees, so the traditional kind of pollinators. So it's important for us to realize that when we go out and we look at pollinators, we should be considering these other groups of pollinators because they can often be a little bit more resilient to climate change, and they can often be access other resources that traditional bees cannot. And I'll show you a figure in the next slide that kind of has to do with this. But generally, the Doyle et al. Review, it made a lot of waves in the community because they looked at, you know, about 93% of crops that they looked at can be pollinated by Hymenoptera, whereas 72% can be covered by Syrphids. That's, you know, that's a pretty amazing capacity that we really don't appreciate. People here don't really know a lot about Syrphids that live in BC, and often we kind of overlook them when we're talking about pollinators. The figure on the right is from a study by Rader and colleagues. This is another big meta-analysis. They looked at 19 crop studies. And what you can see there is they looked at the increase in pollination relative to honeybees when you have other bees present and also non-bee pollinators present. And by non-bee pollinators, we're talking about mostly flies, but also butterflies and moths to some extent. And so when you add these to the community, or when you maintain these in the community, as we up here are very lucky to have lots of native pollinators, if you can keep those pollinators around, you're going to experience much higher pollination success than if you're just relying on honeybees because all the other pollinators have taken off, or, you know, conditions are not good for them or whatever. If you can maintain those communities there, you're going to see pretty large benefits in terms of pollination success and fruit sets. So in terms of yeah, what they're doing. But the story is not just about the number of species you have out there on the land. So you can see this is a preprint, so it hasn't been through all the way through peer review yet, but this is data from somebody I really respect. Bartemius is one of the co-authors on this paper. They went out and they looked at how richness within a given taxon. So if you just have a lot of different bee species that are all closely related, they tend to do similar things and they interact with similar kinds of plants. And so richness alone, when we talk about diversity, richness alone, having a lot of species is not always what we should be going for. The key feature here that's going to help increase crop yields and increase pollination success is to actually have functional diversity in pollinators, which means that we need to have lots of different kinds of pollinators. So having high pollinator biodiversity, we need to think of that as having, yes, lots of different species of bumblebees, but also species of andrenids, species of megacylids, and hoverflies, and broad taxonomic diversity. Because you can see on the right here, in those two figures on the right, they show that when you have a lot of functional diversity in your pollinators, so lots of pollinators that do different things and can interact with different kinds of flowers, your yields go up and you see that expected benefit from pollinator biodiversity. And that's one thing that when we go out and we quantify biodiversity, we need to start thinking more about not just counting the number of species that are out there. We also need to think about this broad scale phylogenetic diversity. So how many different kinds of hoverflies, how many different kinds of bumblebees, how many different kinds of mining bees? Lots of broad diversity should be what we're aiming for here, because that's what's going to get you the benefits in terms of pollination. And the reason for this is because pollinator taxa, the kind of less closely related ones, often have complementary habits. And this finding comes from a study by Kusser and colleagues that came out this year. It's really actually amazing. They studied 36 cotton fields in over a 3-year period. So these findings are really good. They looked at the insects visiting individual flowers on plants. And they actually found that butterflies and flies and bees all do slightly different things when they're out in the field. So flies tend to show up earlier in the season than bees. Butterflies and flies access different areas in the plant, so they can access different flowers on the plant than bees can. And as a result of this diversity among different taxa in the kinds of flowers they visit and the time of year and time of day that they show up, they showed that butterflies and flies essentially increase your crop yield by about 50% relative to what would be there if it was just species of bees that were visiting plants, which is really pretty amazing and should kind of make us think more seriously about what we're talking about when we're talking about pollinators, and the fact that we should be monitoring these alternative pollinators. We should be thinking about them as a resource and something that we can manage for, because without them, you know, they estimated something like $120 million US would be lost every year on the cotton crop in the States without kind of those two alternative groups of pollinators. And kind of one of the supporting findings here that one of the studies that prompted this inquiry by Kusser et al. Was this kind of older study by Lysenkov, where he went out and he looked at how far different kinds of pollinators travel in agricultural areas, and how effective they were. And he found that dipteran species, so flies, hoverflies in particular, those top blue lines, Aristolis arbostorum and Sphaerophoria, they tend to travel farther and still effectively pollinate than, you know, kind of our more traditional bumblebee species, which are the bombus species, or butterflies, Pyrrhus brassicae, which is like a cabbage butterfly. So I think this is kind of really interesting because it tells us that there is probably adaptation happening within systems already. We know that there are hoverflies out on the land. They're probably already pollinating things that we don't know that they're pollinating, but we haven't really spent much time tracking them or quantifying them very effectively. And we need to start thinking about this because ensuring high biodiversity, particularly this broad phylogenetic diversity, might ensure against climate change induced pollination declines. So we know that changes in plant timing and extreme weather can reduce pollination success. There's lots of evidence that when the flowers bloom at the wrong time, if there aren't enough pollinators present, often you'll have a pollination, kind of a limit on pollination. And so if we can build pollinator diversity and we can ensure that there is a lot of functional kind of diversity out there, we can build resilience in pollination services. And so a good example of this is from a study by Bartemius et al. Those are different families of bees, so different Hymenoptera families on the right. And you can see that center kind of dotted line, that indicates kind of the typical apple bloom in the eastern US over a number of years. And what they did is they went out and they looked at when these different families of bees, or when these different genera of bees emerge from their overwintering, and so are available to pollinate apple orchards. And they found that generally, regardless of when those apples bloom, there's usually something there to pollinate it, because these different genera of bees have different emergent states. And so this is kind of another demonstration of that nice functional diversity, right? So different emergent states equals there's always something around to pollinate. So if we can preserve this kind of broad kind of variety within the pollinator community, usually you probably won't have problems with pollination. So in terms of the kind of reasons for establishing a baseline here and now, climate change is accelerating, we know that. What you also may not know is that very little work has been conducted on pollinators anywhere between kind of the Okanagan and the Arctic. So there's lots of research that has gone on in the Arctic because climate change is proceeding very quickly there. It's a huge site of interest. There's been some work in the Okanagan because traditionally lots of agriculture has occurred there. But in between those areas, there's a big blank space, at least in Canada. We know that growing seasons are starting earlier and lasting longer. And the fact is, people who produce food, people who grow stuff, might be able to take advantage of this if we can make sure that the pollinators are there, or if we need to manage more effectively for those changes in the seasons to ensure pollination. This might be the time to be able to establish that baseline and kind of prepare for the future. So in terms of how we're quantifying diversity, generally, yes, we go out, we count how many different species do you have. That's your basic biodiversity measure. It's just called species richness. So you can say we have 2 different species of flies, 1 species of butterfly, and a couple of species of bee. But in a more advanced kind of way, and this is what we're sort of working towards, and we should be able to get a little bit of a handle on it. Ideally, what we want to start doing is thinking about characterizing communities. So one of the things that the data we're collecting right now is going to allow us to do a little bit is to look at relative abundance of different species within a community. So is the community really dominated by one particular species of pollinator, or are there similar numbers of a whole bunch of different pollinators? More advanced work, and this is work that we would have to go out and do surveys on vegetation and watching flowers, literally observing what visits flowers, would be to start looking at plant pollinator associations and building networks. So network modeling, you may have heard of it. And then kind of the most kind of complicated way to think about quantifying diversity is to look at different functional roles and kind of niche occupancy. So what sort of plants are available? Are there pollinators for all those plants that are available? And so on. But all that we're really able to do right now with this kind of basic method that I'm going to talk about for a sec here is to quantify species richness and a little bit to look at species evenness, so the relative abundance in the communities out there. So what we did over this past summer, we recruited 18 sites between Dunster and Talquah, and so they're all active producing farms. And at each farm, we had kind of 2 different kinds of traps that we set up. There are vane traps and there are pan traps. And the way that we've targeted pollinators is to use color. So all the traps have different colors in order to attract pollinators more specifically than just being a big net out there. We sampled at about 3-week intervals between May and August. That's a map of the sites. You can see it's a really big area. We covered a lot of turf this year. Lots of different production systems. So about 5 of the sites are kind of pasture and ranch production. 6 sites were forage crop production. Most of those are alfalfa. And then we had kind of mixed vegetable and fruit production on 7 sites. Within that group of 18, 2 of them are certified organic, but none of them use any kind of spray applications of herbicides, pesticides, fertilizers. So it definitely doesn't reflect the kind of traditional, big monoculture, big ag model. And if we do a second year, maybe we need to expand into that, but it's just something to kind of keep in mind. So vane traps, this is what they look like. I usually would set them up on the margin of a field, right where it backed onto some kind of forested area. Blue, because blue generally attracts a lot of bees, and they've been really effective. We caught lots of bumblebees, fair number of flies, especially syrphids, and butterflies and moths. The pan traps are these little bowls that are painted with kind of UV paint, so you can't really see it, but that white bowl on the right there actually kind of glows in the dark to insects because they can see in the UV. And I did transects of these, so usually on a given site, I'd put some in on the interior and some on the edge of field. And they're generally pretty effective for lots of flies and many solitary bees, which are not as effectively sampled by vane traps. In terms of the progress thus far, this was accurate like a week and a half ago. We're somewhere in the neighborhood of 7,000 specimens at this point. And what I've been doing is pulling out samples, and we've been sending them to be DNA barcoded. So a small portion of the genome gets sequenced, and that helps us identify specifically what species is this particular specimen. And so right now, I think the count is at about 274 kind of potential pollinator morphotypes, and that's going to grow. So in terms of the barcoding results, a good chunk of what we got out of that first round were known pollinators. And I wanted to draw your attention in particular to, there's a couple of groups that you need to start keeping an eye out for. The megacylids and the andrenids. These generally require sandy soils and of course woody debris in order to nest. And so this kind of leads me into, when you're thinking about managing for pollinators, what do you want to do? Well, landscape simplification generally reduces pollination. We're quite aware of that. And so any way that you can retain edges and semi-natural habitat on the land is often a really good idea. The figure just on the bottom here is from a paper by Martin and colleagues. And you can see that there's kind of a sweet spot where if you could retain some semi-natural area, that increases the edge density, so that those kind of two interfacing ecosystems, you've got your agricultural land and your forest. And in that little kind of slice of land, that's great in terms of increasing pollinator biodiversity. The other thing to really think about here is that tons of studies show that plant diversity equals pollinator diversity. I'm running out of time, so you can just see the line goes up. It's a pretty clear positive relationship. In terms of how you can maintain and promote floral diversity, well, you can retain floral resources by avoiding mowing. If you don't mow stuff, the flowers stay on it. The resources are there. There's been an initiative in the UK. You can see the little icon on the left there called No Mow May. And that's kind of in that vein. The other thing you can do is manage margins and non-productive areas differently. So in terms of those bees that need sandy, dry areas for nesting, don't go in and dump a bunch of stuff on top of that, right? Like leave some of these margins in these quote unquote non-productive areas, leave them be. There's stuff growing there. There's often flowers there. And the other thing too is to think carefully about timing. So particularly with mowing, early in the season is often a pinch point nutritionally for pollinators. So if you can hold off on mowing floral stuff until June, July, often that'll give them a chance to get going in the spring. Okay, I'm going to jump over that, but we're working to finish the project. I am putting together some stuff for the Entomological Society of Canada meeting, so we'll have some information. I'll probably send the materials on to Harmony to post for people who are interested in kind of the more academic part of this. Yeah, I'm currently looking at some geographic and temporal trends, but we should have some more information to CAI in terms of a final report and kind of more quantitative findings by January. I'm still working through some samples, but we're getting there. And if you're looking for more resources, I wanted to recommend a few. There is, the first two are databases where you can put in a pollinator name or a plant name and get back a list of all the plants and pollinators that it interacts with. So if you're thinking about growing something new, or you have a name of an insect where you've seen it on your land, but you don't really know what it does, what you can do is go into these databases and look up and see if there is a potential pollinator that you have out there already, or perhaps if you're looking for a plant in order to encourage a particular pollinator, go to these databases, put in the name of the pollinator, and get back a list of stuff that you potentially could plant. They're really kind of great. The Insight Guide is a guide to bees, pollinators of BC. It's from down south, but it is quite good in terms of the diagrams and kind of some of the information it provides. The Surfids of Canada guide, that's for hoverflies in Canada. It is highly technical, but the pictures are great. If you're looking to identify something that isn't a bee, but looks like a bee, I would start there. And then there's a great lab for pollination ecology at SFU that you can use if you're looking for some information. They have some pretty accessible stuff there. That's the L lab at SFU. So yeah, that's where I'm going to leave it. I think I'm still within my time slot. I'm going to take a look at the Q&A here.
Yeah, there's 2 questions in there, Aija. So, 3
Yeah.
now. And certainly if anyone has any else, please feel free to put them in. I'm not sure if we'll be able to get to all of them. Oh, with these 3 we can, but if there's lots more.
Okay.
But I'll maybe let you, I don't know if you want to start at the top and just go down.
Yeah. Okay. So
Yeah.
in terms of how do we effectively monitor our diversity, given the large diversity of pollinators and collective limited knowledge of taxonomy, role, niche use, etc. So yeah, the fact is that this is where tools like DNA barcoding and actually employing actual people to go out and do this work. Like I'm sorry, but to a certain extent, if we're going to really properly monitor diversity, you need to have a really good specimen ID, right? And the fact is, the only reason I am able to do this gig is because DNA barcoding exists. I am not a taxonomist. I am not a systematist. You know, the DNA barcoding really is a powerhouse for things like this, because it means that somebody like me who has like a moderately, like I've been sorting bugs for a long time. You know, a big chunk of my PhD work was on sorting these massive insect terrestrial arthropod samples, but I'm still not getting stuff to species. And so the story is for somebody like me who's moderately skilled to be able to separate things out into, okay, I think this is a distinct thing. Let's send it off and get a really good, strong ID from the DNA. That really kind of strengthens our ability to do this. So I think number one is, yeah, you can't just say there's 5 kinds of bees. You do have to spend the money and you know, get the data back on what in particular, what species this is. But the fact is that generally there is a fair bit of good information out there about life histories and kind of the basic naturalist information for quite a lot of species. We have a decent sense of it. So we know generally, you know, that if it's a hoverfly, it probably has a larva that eats aphids, right? We know generally what its life cycle looks like. More specialized studies are probably something that need to be undertaken by academics, like in terms of really studying one thing. But at the end of the day, at least if via this project we can get a fairly decent list where we can say we've got, you know, 20 species of hoverflies in the region. Here is where they occur. I think that's a good start. I know it's the kind of thing where how long is a piece of string? Like we're never going to know enough about a lot of these species because, you know, they're cryptic or they're hard to find or they're only present for a short period in the year. But at the very least, by having a list that's an inventory, so we know at least what to expect on a given site, You know, the kind of the what's out in the published literature already in terms of our understanding of their basic life cycles, the kinds of things they eat, you know, the sorts of flowers that they interact with, we have a reasonable sense around what we can do to help encourage, you know, populations of different species and kind of keeping, yeah, and kind of keeping biodiversity where it's at, I think. Okay, so the next one. Let's see here. Highlight the advantages. Yeah, Harmony?
Oh, Asia, I was just gonna say, you know what, we're running out of time. So maybe if
Yep.
you want to pick the one that's best to answer live, and then maybe the other ones
Okay.
we can you can type in an answer while
Yep.
Bonnie is presenting, if that's okay. I'm so sorry.
Okay.
We just
Yep.
only have like 5 minutes for
Yep.
Q&A. Awesome.
So in terms of, perhaps I'm not reading the graph correctly, but does pollinator richness really decrease fruit set? Well, that is from regression. So it's controlling for all other factors. And it hasn't been through peer review, but what they're showing, it's not a causation, it's a correlation. So yes, fruit set in these systems where McGrath and colleagues studied it, they saw when you just have really high richness, but it's all within a given taxon. So it's a very narrow, it's richness, but it's controlled for functional diversity, right? So functional diversity is essentially 0 on that graph because it's from a regression. So it's a statistics thing. So generally we would expect functional diversity to increase as richness increases, but because of the statistics on the way they put the graph together, it's held at 0. So if all other factors held equal, as richness increases, basically the community just gets more kind of dense instead of having high functional diversity. So high functional diversity would be when you see fruit set increase. Usually richness and functional diversity are correlated, so they would go up together. But the way the stats were done on that paper, if you take out the functional diversity part and you just look at how many species are there, species alone is not a great predictor of how good the pollination service will be. So I hope that answers your question. I will type in some answers to questions while Bonnie gets going here, I think.
Yeah, Bonnie's up. Thank you, Asia. Thank you for those answers. And yeah, Asia will answer you as best she can over the Q&A. So I will introduce our next presenter, who is Bonnie Zand from Bonnie's Bugs IPM, a Vancouver Island-based consulting company which assists growers with managing insect pest issues. Bonnie is currently the project lead for the Vancouver Island Pest Pollinators and Beneficial Project, which is working with Vancouver Island producers to increase IPM knowledge and implementation in the region. Welcome, Bonnie. Thank you.
Thanks. Is my screen showing up all right?
It is.
Perfect. All right. So hi, everyone. My name is Bonnie Zand, and I run the Vancouver Island Pest Pollinators and Beneficial Insect Project. I was on the webinar last week where I talked about how our project is helping to increase pest monitoring and IPM practices on Vancouver Island farms. And this week, I want to talk about how our project monitored for pollinators and other beneficial insects in agriculture using a citizen science platform called iNaturalist. I want to start with a brief explanation of citizen science and iNaturalist, and then I'll talk about how we set up our project and the types of data we were able to collect. So to start with, what is citizen science? According to National Geographic, citizen science is the practice of public participation and collaboration in scientific research to increase scientific knowledge. Through citizen science, people share and contribute to data monitoring and collection programs. So citizen science allows our project to collect data about the pollinators and beneficial insects present in agriculture by harnessing the collective observations of farmers, gardeners, agrologists, and naturalists, rather than doing what Asia was doing and going out ourselves and collecting the data using pan traps and blue vein traps. As well, our citizen science project encourages engagement by the collectors with the data. And so that's also a really valuable part of our project. And so how do we do citizen science? There are many different models, but we used a platform called iNaturalist. So iNaturalist is a worldwide social network that's available to anyone with a camera and an internet connection. And it collects and shares biodiversity information such as occurrence data and identifications. And so here, if you were to go onto iNaturalist, this is what you would see. Each of these red dots is an observation that's been made and added to iNaturalist. You can see it really is worldwide. And thus far on iNaturalist, there are over 83 million observations have been made of 345,000 plus species. And those observations were made by almost 2 million observers. So that's an awful lot of data that is available to scientists to use and to look at. So how does iNaturalist work? So in order to use iNaturalist, you need to get a free iNaturalist account. iNaturalist can be used, there's a desktop version, as well as there's an app for your smartphone, either iPhones or Android. So a person who is an iNaturalist user is going about their daily business, and they go out into nature, or in the case of our project, they go out into an agricultural system and they see something that they would like to make an observation of and add to our project. And for our project, we really wanted to make it really broad and to not have needing to know about pollinators and beneficial insects in advance be a limiting factor on who could contribute. So for our project, we said we were accepting observations of any insects found in agricultural systems. You didn't have to already know what it was or that it was a beneficial insect. So one of our observers sees something, they think, hey, this is something in my garden, I'm not sure what it is. I think this project might be interested. So then they need to make an observation. And so the first thing they do is they need to collect evidence of what they're seeing. And in the case of our project, that evidence was a photo. So you take a photo of the insect you're seeing. You also need to record the date and the general location. And again, if you're doing this on your smartphone app, it will collect that date and location data automatically with the photo. So it's very simple and straightforward. When that user then goes to upload that observation onto the iNaturalist platform, they'll also be asked to make a suggestion for what they think they're seeing. And again, this needs to be really accessible, and it is. So iNaturalist has a computer vision model that will compare your photo to other similar photos and provide suggestions of what that thing in the photo might be. And so this is one way that this platform both provides information to science and also gives back to the people that are using it. Quite often, if it's a common species or a good photo, you can get a really good identification just by uploading this to iNaturalist. But sometimes that computer vision model is wrong, or the photo isn't quite good enough. And in that case, the user just puts in their best guess at what they saw. And again, this does not have to be anywhere near the actual species name. So you can say, I think it's an insect, or I think maybe it's a bee, or I just think it's an alive thing. And that's good enough. And then the social networking aspect of iNaturalist kicks in. And so additional iNaturalist users come along and they add their identifications onto that observation. And as additional identification suggestions build up, the identification becomes more specific, and eventually the community arrives at an agreed on species identification. And at this point, this observation attains research grade status. And that gives any scientists that are coming into iNaturalist and using the data some extra confidence that those species identifications have been verified by multiple people. So it's a pretty powerful tool. And I want to show you some of how iNaturalist can be used. So I am going to, if this works, we're going to go. All right. So this is, again, I've gone into the iNaturalist page and I'm looking at iNaturalist observations on Vancouver Island because that's my region. And so here you can see these are all the observations that have been uploaded to iNaturalist from Vancouver Island. And if I'm specifically interested in a group, like for example, bees, I can type this in. Let's see if it's going to do it for me. Thinking, here we go. So now iNaturalist is bringing up all of the bee observations from Vancouver Island. You can see we have almost 10,000 observations of bees, and 87 different species have been recorded from Vancouver Island over iNaturalist. You can also see in this map one of the issues with using iNaturalist, and that's that these iNaturalist surveys are being done by people going out and spontaneously deciding to add their observations. It's not systematic. And so even though there are very few observations here in the northern and central parts of Vancouver Island, I promise we do have bees in the northern and central parts of Vancouver Island. And so this is one issue with iNaturalist, is that the data is quite biased towards things that people see and decide to take photos of. The bees. And then another issue with iNaturalist is that while it's collecting this data, and we've got, we have the locations, we have the photos, and we have the dates, which allows us to look at all the different things. iNaturalist on its own is not very good at collecting data about specific habitats. And so for our project, we really wanted to know not just what pollinators are present on Vancouver Island, but we wanted to know which ones are present in agricultural systems. And so in order to do this, we created our own project on iNaturalist. And so this is our project, the Vancouver Island Pest, Pollinators, and Beneficial Insects Project. And this was a way for us to collect observations specifically about agricultural systems. And so anyone can join this project and look at it. And any observations added through our project are also part of the overall iNaturalist data set. But when people add observations specifically to our project, so they click add observations, and they come up to this screen. And so this is the general data upload for a desktop version of iNaturalist. So this is all the where, when, what information that all iNaturalist observations have. But for our project, we also got to add these specific project observation fields. And so this first one here is the most important one. And so this is where we are asking all of our participants to tell us what the observation location was. And in this case, we're actually talking about agricultural scale. And so people are able to choose, did they see this insect in a garden, a hobby farm, a small, mid, or large scale farm, or was this observation made in a non-agricultural setting? And so this is allowing us to filter our results and try and focus on things seen in agriculture instead of things seen in parks or ski resorts or beaches. We're also asking for a little bit more information because, as Aija was talking about, we're interested in plant-pollinator interactions and what types of crops and what types of systems. And so we're asking growers to tell us the type of crop. And we also ask a question about what type of farm practices are used. Whether this is a conventional farm, an organic farm, or perhaps the user doesn't know. There's also a spot to add some notes with more info. So if we go back here to our project page, as well as letting us access this additional information we're able to ask about, having a page also helps us to engage with producers and with the people that are doing our observations. And so we're collecting the data here. And so anyone can come and look on this page. And so they can have a look. We look at our stats. So far, we have 825 observations of 277 different species that have been observed in these agricultural systems. We can have a look at what the most observed species are. Our top runner right now is the yellow-fronted bumblebee, followed by some lady beetles, the Vancouver bumblebee, and the European paper wasp. This project page also has a map. This map has all of observations, and so you can see where they are spread out. And so again, definitely some observer bias here. We've got a lot of observations in the Comox Valley. We also have quite a few in the Capital Region and the Cowichan Valley. And we need to do some more outreach to get some more observations in other parts of Vancouver Island. But for producers, they're able to come and look at this map, and they can actually zoom in on where their farms are and look around and see what's being observed on farms near them. Our project page also allows us to engage with our community of observers and producers by producing journal posts. So here we've got one on beneficial beetles, and these journal posts allow us to highlight some of the observations that have been made through our project and share that information out, provide a little bit of additional information about, in this case, ground beetles or other beneficial insects and pollinators. So we can have a look. If we wanted to check out and see what we've seen, we can look at our species list. There it comes. Oh, it's coming, I promise. There we go. So we can see 825 observations of 277 species. And so this is our complete species list. And for each species, it will tell us how many observations we had of that species. So we have 22 observations of the yellow-fronted bumblebee, 17 of Asian lady beetle. And as we scroll down, you can see each of these species that we have observations of. And for any growers who are interested, once they put their observation up, they've gotten that identification back, the communities come in, they can go and they can click onto the species page for any one of these species, which is going to pull up some extra information about that species, like its seasonality. And it also provides more photos for identification and links to things like Wikipedia articles, so you can learn more. And I'll just show you one specific observation just to show you what that looks like. So this is an observation of 7-spotted lady beetle. And this observation was made by user Jay Rosinski. We can see it was observed on June 10th, 2021. And again, we've got the map that shows us that this was in Foria region. And here's the photo. And so we can see here are our lady beetles. And one of the advantages of using iNaturalist is that we are getting these photos, which show us a little bit about the habitat and the plant interactions that we're seeing here. So we can see here, these are on a fennel. And if I scroll down here, here's the identification activity. So we can see the initial suggested identification was that this was a lady beetle. And then you can see a number of other people have come along after that and said, it is a lady beetle and it's a 7-spotted lady beetle. And so you can see we've got 3 different confirming observations there. And so this is a research grade observation of 7-spotted lady beetle. And another aspect is the social aspect. So we can see that users can have comments and there can be a little bit of discussion here about what's happening in this observation. And so that's also valuable for producers who are perhaps trying to decide what plants they should be growing to attract different beneficial insects or pollinators. And then over here, again, these are our observation fields that give me a little more information. So I know that this crop type was vegetable and this observation was made on a certified organic small scale farm. And so that's sort of a summary of some of how our project works and some of the different things that we can learn using iNaturalist. And I'm gonna flip back to our PowerPoint here and just finish this out. So some of the important things that we learned going through this process of having this iNaturalist way of getting this data. One of the big things is that outreach is really important, especially because we had a specific project within iNaturalist and needed our observers to go through that extra step. It was really important to get the word out about our project and to have growers join. And so we did outreach through our grower IPM newsletter that we sent out with the rest of our project, through our Facebook page. And we also did outreach to gardening groups and naturalist groups, trying to get people who are out and observing in agricultural systems. Another thing we learned is that growers are really busy. And we were launching this program in the middle of the growing season. And so we did find that more than 50% of our observations came from gardens and hobby farms, and not so many from the mid to large scale farms. So one of the things I'm hoping we'll do this winter is do some more outreach in the off season when growers have a little more time. In addition, we saw that data can be a little bit hard to interpret. There is definitely user bias here, and some groups are observed more frequently just because they're more obvious. We also found that small insects were hard to get IDs from on photos. And so a lot of our larval insects or small bees or small pest insects didn't have very good identifications. And as well, not all groups have access to the same expertise. So there are some world bee taxonomy experts that use iNaturalist. And so bees typically are quite well identified. I haven't found an aphid expert that's been coming through and identifying our photos yet, though. And a big advantage of the iNaturalist was that those photos did provide a little bit of additional information about habitat and floral associations. So I hope that gives you some ideas of how iNaturalist can be used to do this type of work. And anyone who's interested, please join our project. And if you'd like more info, my email address is here. And I just want to say thank you to everyone who helped out with our project. And I'm ready to take questions.
Wonderful. Thank you, Bonnie. Well, we've got 2 questions here. So I will start by reading those out. We do have a few minutes. So if anyone else has a question, please do add it into the Q&A and we'll do our best to get to it. So the first question, does iNaturalist rank identification by observer? Some of the users would be closer to expert identification and other users might be less reliable. And often the crowdsourced answer is correct, but not all observers are well trained. So I guess, yeah, getting to that question of how are those observations vetted, and, you know, a really interesting contrast to Aija's work, which is, you know, Aija is collecting and sorting specimens and using DNA barcoding. So we've kind of got the 2 extremes there.
Yeah, so this is very definitely not DNA barcoding. And the question is correct. Some observers are much better than others. And so this is where that community aspect becomes important. People can put up incorrect observation identifications, but then hopefully other users will come along and say, no, that's not what that is. And so it is required to have a consensus. So it can't just be one person putting it up and saying, oh, it's this. You have to have at least 2 confirming identifications. But it's also important for any scientists that are going in and using that data to keep in mind that these identifications are being done by people who are not necessarily experts. And so you definitely have to take it with a grain of salt, and especially for identifications. You know, sometimes someone will put in an identification for something that, you know, is a European species and doesn't occur in Canada. And so that would be a big red flag that you might want to send that photo to a real expert and get that double checked before you add that particular species to your list. But at the same time, the identification function, one of the things we really liked about it was that it did allow the growers to find out what was happening in their farms. They could put those identifications up, and, you know, even if they didn't come out perfect, you know, just knowing that, you know, that little green thing buzzing around is actually a bee. It could be really useful for growers to be like, oh, hey, I didn't know. And so it has disadvantages for sure, but there are also advantages. Other questions?
That's good. Well, on that note, we actually have a project that will be starting up in the Balkan Chaco Fort George region that's actually going to use both. So using DNA barcoding and citizen science. So it'll be interesting to kind of have a project that will be utilizing, you know, those 2 very different methods. So that will be starting up for the next growing season, looking at pests. So the one last question here before we wrap up, just from just a question about, are you interested in collecting data on the Southern Gulf Islands?
We would definitely accept observations from the Southern Gulf Islands.
That's great.
In our project. Yeah, where we're considering Vancouver Island to also include all the Gulf Islands. So yeah, we're definitely love to have people join and add their data.
Wonderful. Well, thank you so much for that presentation. Thank you, Aija. Thank you, Bonnie. That was really wonderful and so interesting to hear those different approaches. It is the end of time. So, you know, as we've discussed, these presentations really summarized these 2 different methods that are being used, but both to develop that really important piece of information that we don't have for British Columbia, which is on the baseline data, developing that baseline data for pollinator populations, which we know, you know, those pollinators are very important for all of our agricultural production. We certainly do hope that similar baseline data projects will continue to happen or will get started in other agricultural regions of the province, and hopefully some of these methods can help to inform those projects going forward. And as always, and Aija spoke quite a bit to this, you know, it's really important to also then develop some producer focused resources, some management resources about habitat creation and retention to really assist producers with ensuring that they're doing what they can on their properties to help pollinators survive well into the future and ensure that they're there to pollinate our crops. So thank you so much for joining us. Thank you, Aija and Bonnie. I will, if you want to keep up to date on project results, events, new resources, you want to know what happens with Bonnie and Aija's projects, you can subscribe to our e-newsletter. I believe Shauna was going to enter that into the chat. So you can have that link available to you. It's also just on our website. We will be sharing the recordings along with the roll-up of the resources mentioned during today's session, as well as registration links for the upcoming sessions. We do have our next session is coming up next Tuesday, November 3rd. I believe that's Tuesday. Investigating crop suitability for changing climate solutions. For changing climate conditions. And then November 9th, we have agricultural water management in a changing climate. So thank you all. Have a great rest of your day. And hopefully we'll see you on November 3rd.